Hello everyone, my name is Nina and I am a master's student in the Garrett lab, and I'm going to be talking today about my master's research, which looked at using airborne eDNA to survey tropical bat roosts. So a very brief introduction, for those of you who maybe didn't see my supervisor's talk on Sunday, airborne eDNA for at least terrestrial vertebrates is a very new, only come about in the last few years. It's primarily been used in the medical field for things like pathogens, microbes, with a little bit of work in the realm of plants. And so I wanted to take the method that came out of a couple of my supervisor's papers and see if we could deploy it in the field to survey bat roosts. So this is what our samplers look like. The way it works is it's got a computer fan on the bottom that pulls air through a furnace filter that has been suspended above the actual fan using a custom 3D printed attachment, and then it's held in place with that ring. We treat those filters as like a tissue, so then that's what we would extract the DNA from. We amplified using 16S mammal primers, and then sequenced it and ran it through BLAST. The first sort of step that we did was we went down to our field site in Belize, where we have this sort of field lab where other researchers who are working on the bats themselves will bring bats back that they've caught in the field to be processed. And so that's what you see on the bat line over there. Each one of those bags has a bat in it. And the really nice thing about this is we know exactly what species are coming into the room and how many of them there are. And so this kind of acts like an artificial roost where you have a mixed species community with different turnover rates each night. So that was where we first thought we could deploy and see if this was something that would work. It worked quite well. So we detected our very rare species like the mormoths, which we only caught once throughout the whole 2-week period. Even amongst the very common species like our Glosophagus species, which we were catching maybe 30 of them a night. So what we get from this is that sorry, I thought that was the microphone. What we get from that is that airborne eDNA can characterize very complex species communities with mixed abundances and mixed species. So the sort of natural, not the natural next step. Okay, so we also, because we had this room where we knew the exact abundance, we also ran a quick correlation between recount and cumulative species. There was a general positive correlation, of course. Using eDNA to detect abundance, as we all know, has its pros and cons. And of course, there were these outliers. So we have these 3 up here and this one down here. So the 3 species at the top, they all have, there's actually 2 species in that genus at our field site. And most of them are very difficult to tell apart. So Glosophaga, for example, you can only tell the 2 species apart by looking at them very closely and looking at their teeth. So every time one of those bats was brought back and it was labeled Glosophaga, it was taken out of the bag, looked at, put back in the bag to confirm species ID. The Desmonas rotundus, on the other hand, the common vampire bat, you actually don't even really need to open the bag to know. You can kind of just touch it and it usually hisses at you and it's like, oh yeah, that's a vampire bat. So what we think was going on here is that the more you handle the bat, the more DNA shedding you're getting. So while there is this sort of positive correlation, obviously there are other factors impacting shedding rates. We also got some kind of outside detections that we think came in through the window. So things like howler monkeys, kinkajous, climbing rats, which we've seen in the area and often heard above that field lab. So these are things drifting in through the windows. And so that's kind of something that we need to think about, what's being brought in on the wind. We also had instances of secondary transfer. So these were very, very low positives, like 1 or 2 recounts of species that didn't even occur in Belize, like they were African bat species. And we traced them down to some of our colleagues who had tracked in this very small amount of DNA on their equipment. They've been working with those bats months previous. So it kind of makes you rethink contamination a little bit. So the natural next step to that was then to deploy our samplers in natural roosts. to see if we could actually detect bats in their roosts under natural field conditions. And so I deployed my samplers in roosts like tree roosts like this, as well as a few others, which I'll talk about in a minute, in the archaeological reserve, the Lamanai Archaeological Reserve, the Kakadish Archaeological Site, and then a few others in the Indian Creek area of the Orange Rock of Belize. So we kind of had 4 main roost types. So one was those tree roosts that I showed you. The other were natural caves like this. This is what I'm going to be referring to as the schoolhouse cave from now on, and I'm going to come back to it in a couple of slides. The next was Mayan tunnels that had been dug into old Mayan ruins at the Kakadish site by both archaeologists and looters. And then the final one was other man-made structures, which were things like an attic and an old cistern. And in these roosts, some of them we knew bats were roosting in, so you can see a couple over there. Others we thought we just suspected. So it was kind of like a confirm that the bats are there in the places that we know they're roosting, but also check and see if they're in any new roosts. For the majority of our roosts, we placed our samplers, which you can very nicely clearly see here, can be battery powered, inside the roosts. There were a couple instances where we couldn't do that though. So for all the tree roosts, one of the caves and one of the tunnels, we couldn't actually put them inside. So the tree roosts were often either too high or the entrance was too small for us to go inside. So we just kind of hung them in, or in one case taped it to the outside. For the tunnel that we couldn't get into and the cave, it was considered unsafe to go inside. So in that case, we just placed them outside with the idea that as bats were emerging, they would be flying over them. We sampled for up to 24 hours. There were a few instances where we could only sample for about 6. So for those tree roosts, they were on the archaeological reserve, which was open to the public, and very kind staff who was letting us sample there asked that we not be there while the public was there. So we, of course, were like, oh, sure. So it limited the time that we could access, but it ended up being at night, which, as I'll go over in a little bit, might have been beneficial. It worked remarkably well. So we detected 11 bat taxa, several non-bat mammals, and a couple of amphibians. And this was really exciting for a number of reasons. So the first is, as we kind of heard in the talk before, bat roost surveys can be particularly difficult, especially in the tropics, where a lot of the species don't make species-specific echolocation calls. This is also a potentially non-invasive way to do it, so you're not actually handling the bats. That can not only limit the disturbance to the bats, but also help stop the spread of human-bat disease transfer, as well as disease transfer between bats as you're handling, which, when you think of things like white nose syndrome, could be a very important thing to consider. It also means that you could survey roosts potentially semi-remotely, so you could go into one roost, set up your samplers, leave, go into another roost and do the same thing, and potentially sample more without necessarily needing to increase your field effort or the time and resources that you would need to do that. Most exciting, though, was our detection of the white-winged vampire bat. So this is a species that has always thought to be in the area, but in over a decade of going down to this field site and doing annual surveys, had never been confirmed using traditional methods like mist netting or acoustics. They can be particularly difficult to catch using those methods, and we managed to get it on the first try with airborne eDNA, so that was very exciting. And what we get from that is that airborne eDNA can find missing species, and that's often something that we see in more well-studied areas of eDNA, like aquatic eDNA, as being one of the main benefits of using eDNA. So I talked a little bit before about the school cave. So we had more access to this cave. It was also one of our larger caves, so we ended up with kind of 2 sets of samples. One that was taken during the day and one that was taken overnight. And what we noticed was, kind of anecdotally, in our data, we had more detections at night for bats than during the day. It was not significant when we ran statistics. Our sample size was really small because we didn't even know if the method would work at all, so we weren't really planning ahead for this kind of stuff. But we want to go back and do it again because, as was kind of mentioned in the previous talk, bats are more active at night, so it could make sense that we're detecting more of them at night because there's more shedding going on. We had some instances where we detected co-roosting, so this is the big-eared climbing rat. We detected it in a number of roosts, and it's something that people have seen going in and out of the roost. So this is encouraging, as it kind of, not only are we detecting our target species, the bats, we're detecting things that might commonly co-roost with them. Especially interesting is this figure over here. So our one colleague was looking for localizations of the big-eared climbing rat, and she deployed some of her samplers right next to ours. And those spikes in frequency and amplitude are the big-eared climbing rat localizations, but what you don't really see is any other sound really operating in the high frequency or high amplitude ranges. That's really important when you're working with bats because if you want it to be non-invasive, they often call in high frequency ranges. So if you want it to be non-invasive, you better make sure it's not emitting high frequency noise. So this is just an example with and without our samplers. These are actually bat calls. So you can see with our samplers in there or without our samplers, they're still making their calls and their localizations. So even though this was hugely successful, we still have a lot of questions that we don't really know the answer to. So we don't know how many samplers we need to deploy to survey the full diversity of a given airspace. We don't know how long they need to run for. We don't know how far it travels. So we know that it drifts. So if I set up samplers in one roost and go to another that's relatively close, do I have to worry about DNA from that roost drifting into the one that I'm setting up in next? We don't know if the size of the roost matters, and we also don't really know when we should sample. But we've seen a lot of interest in this kind of work in the last few months, so we're very optimistic that these questions will be answered quickly. So thank you for listening. If you have any questions.